So 12 talks about the nervous system, central nervous system. What parts belong to the central nervous system? Brain central, yeah, sure. Brain and the spinal cord, and then peripheral nervous system. Everywhere the outside of it, yeah. Like what? A lot of peripheral. The nerves, yeah, the spinal nerves and then the cranial nerves. Right? You have 12 pairs of cranial nerves. That today we talked about it, and they will talk about it again with Monday's lab students. There are 12 cranial nerves that belong to our peripheral nerves. Okay? So, central nervous system is brain and spinal cord. We can talk about the adult brain. So if you look at different books, they divide it two ways. They'll say 4 parts of the brain or 3 parts of the brain. 3 parts is basically talk about how it develops. About forebrain, midbrain, and hindbrain. We generally concentrate more on the in terms of functional aspects, so we divide it into 4 brain parts. Okay? So cerebral hemispheres, these are the larger portion of our brain. It's also called cerebrum. Okay? And then we have diencephalon in the middle of the within the cerebrum itself, and then brainstem that's lower down, kind of like a tree, and the stem of the tree. And then cerebellum is like a little smaller brain. Right? So this is a cross section where you can see the diencephalon. It's not a cross section, you will see it because it's more internal. Okay? So we below here, and then we talk about hypothalamus. The pituitary gland sticks out of this diencephalon, the thalamus, and the hypothalamus goes back. We talk about in terms of the diencephalon, that word there. Then we talked about the brainstem having 3 parts. The big bump part, that's the more obvious one to identify. That belongs to the middle of it, that's the pons. Above it, midbrain, below medulla oblongata. And then the spinal cord connects to the medulla oblongata. What bony landmark can we find between the medulla oblongata and the spinal cord? Foramen magnum, right? So that's the large opening in the skull. Above it is the brainstem, which more specifically is the medulla oblongata. Below it is the spinal cord. So let's talk about ventricles. So ventricles we do have in our heart. Those are the pumping chambers, but these ventricles are little fluid-filled cavities within our brain. Okay? So we have 4 of them, and what fluid is it filled with? CSF, cerebral spinal fluid. What was this one? Ependymal salt that we talked about on Monday. They do in certain spots, which we'll talk about today. And also, they move the CSF, right? How do they move it? What structure do they have? Cilious. So they're found within the ventricles. They're also found in the central canal of our spinal cord too. Okay. So we have 2 lateral ventricles. We don't call them first and second. We do have a third and a fourth. These are kind of next to each other, so we just call them lateral. They do come very close to each other, especially in front. It's a C-shaped. We'll look at that image in a second. Third ventricle is found within the diencephalon, so mostly within the thalamus. A little bit does go in the middle part of the hypothalamus. And then the 4th ventricle is mainly between the pons and the cerebellum. So we go back to this image. 3rd ventricle will be here, 4th ventricle will be here, 3rd actually will be within the diencephalon, and 4th ventricle in the cerebral hemisphere, so it's more lateral to the 3rd. In terms of fluid flow, so the ventricles are connected to each other. together, they have CSF is actually produced within the ventricles themselves. And the connectors, they keep flowing from the fourth. There are two options. They can go straight down the central canal in the spinal cord, or go to what space contains CSF. It's within the meninges. Who in lab today we talked about that space. What space contains the circulation? Okay, too much attention for the lab exam. It's a subarachnoid space, right? Subarachnoid space is when we think about cerebrospinal fluid, we are mostly thinking about the fluid outside. That's where it is. It is within the meninges. It's a certain area. So, from the lateral ventricles, the connection between lateral to third ventricle is nice and easy. Interventricular foramen. Right? So, it's a duct, it's an opening between the ventricles. The third to fourth is a little bit thicker, longer. That's called the cerebral aqueduct. That connects third ventricle to the fourth. From the fourth, it can go straight down the central canal, or go to the subarachnoid space. So it's actually, there are three openings from the fourth ventricle that can take the fluid to the lateral side, one in the middle, which takes the fluid to the subarachnoid space, which is surrounds the brain as well as the spine. Right? So remembers where the subarachnoid space is located. Subarachnoid space. Which two layers? Between what two layers?
Between the pia and the
Very good. So it's subarachnoid, so it's below the arachnoid, then comes pia. So we'll talk about that later too in a minute, right? And then central canal basically is within spinal cord, so it continues down from the fourth ventricle. This is not from our textbook, but it's a good one from a different textbook, which shows those structures. This is anterior view, so you don't appreciate the C-shape nature of the lateral ventricles, but the third and fourth ventricle, we are able to see that here. This one, the two lateral ventricles are one behind the other, and so one's in front, one's the other. This is the third ventricle, the fourth ventricle, that's our cerebellum, and pons is basically between the pons and the cerebellum. So here would be the interventricular foramen, which connects lateral ventricles to the third ventricle. Then third ventricle to the fourth ventricle is a long tube. That's cerebral tube. And then the fluid can either go down from the fourth ventricle straight down the spinal cord, or come out through three openings and kind of come outside of the brain. It'll surround the brain through the subarachnoid space, which also goes down, surrounding the spinal cord too. So it's actually, as I mentioned, it's produced within our ventricles. CSF is produced within the ventricles. It's called choroid plexus. I think I put that down in our ependymal cells. These are structures with ependymal cells one side, blood vessels on the other side. So it's filtrate of blood plasma. And just do I have the slide about how it goes out? So it does exit back to blood, suprasagittal sinus. We'll talk about it again. That's just those leaves through. So it came from blood, it'll go back to blood. Okay? So it's a continuous circulation. You may have heard that somebody has more fluid, the ventricles that fills up. It's usually overproduction of under drainage. What we do is a shunt from the ventricle to that space, to the vein, venous blood supply is not called the vein, it's called the sinus, to that space, so the blood, the CSF just keeps draining out. It's a continuous process, kind of flushing substances around our brain.
Bless you.
So let's talk about the cerebrum. That's our main brain part, right? One of the 4 parts. It's going to have our conscious thought all from that area. The rest of it's all mostly involuntary control. Intellect is from that too. So we'll talk about the lobe separation. So frontal lobe as intellectual personality and so on. We'll talk a little bit more. We also have somatosensory information coming in. So what does soma mean? Somatosensory versus visceral. From our body, right? From skin, from joints, from muscle, that information also goes. In terms of initiating our voluntary response, it also comes from this. We'll talk a little bit about this part. It's going to be in our frontal lobe. Somatic is in different places. We do have the highest concentration in the parietal lobe, special senses in the other 2 lobes. We'll talk about. In terms of the cerebrum, we do have gray and white matter. What was gray matter mostly made of? Yes, Mondays. What's white matter mostly made of? Myelinated axons and myelinated. So gray matter is unmyelinated as well as large part of the neuron, the collection of the cell bodies. So in cerebrum, it's mostly gray matter. So if you took a slice of the cerebrum, it's gray matter more outside, so cortical, and then white matter more internally. Okay. And there are few collections of gray matter within white matter. And we'll talk about a collection called the basal nuclei. This is in the cerebrum. So any nuclei, another place where they use nucleus, is a collection of gray matter in white matter. So it's surrounded by white matter, there may be little blobs of it. Okay. And for example, cranial nerves, that is a gray matter that starts off and then the white matter comes through. So we'll talk about nuclei. White matter basically centrally located. We do have myelinated axons. What produces myelin in the central nervous system? That's in the peripheral nervous system. Yeah, that one. Oligodendrocytes produces myelin in the central nervous system. Peripheral nervous system is going to be so we'll talk about this in a second. But gray matter is superficial. It's also called cerebral cortex. That region, cortex means outside. So we would say the brain, the cortical region is gray matter. Okay, what was it like in the spinal cord? Was the gray matter more peripherally located? It was more centrally located, so it's flipped in the spinal cord. Okay. While in the cerebrum and cerebellum, we have gray matter on the outside, white matter internally. So let's talk about some structures. So brain is not smooth. Okay, there are some ridges and valleys. Those ridges, the plural is gyri, gyrus is singular. Each one of those bumps out. Okay. The idea is to increase surface area, right? So we talked about in our skin, what structure increases the surface area of the papillae layer of the papillae of the little finger-like projections of it. Dermal papillae. We have some in our stomach too, or in our intestines. Stomach there is a different one. But in our intestines too, we have these foldings, additional foldings, right? Did we know the names of those? Macroscopic and microscopic foldings too? Mm-hmm. Villi and then minor. So whenever we have those projections, the idea is to increase surface area, right? So instead of being like this, we have this much surface area, we have all these finger-like projections, we have these additional surface area. Right? So between the gyri, we have the depressions, the grooves. The shallow ones are called sulcus, so it's the plural, and then the deeper ones are called fissures. Okay? Not smooth, it's bumpy, there are valleys, and then ridges. So ridges are called gyrus singular, gyri plural. The shallow valleys are called sulcus, sulci plural, and then deep one not very visible here. There's a big one here, and the second one's actually between the cerebrum and the cerebellum. Nice big fissures. So we'll talk about the four lobes, then we'll talk about some of the sulcus related to it. So what are the four bones making up our cranium? Frontal bone, parietal,
Occipital,
occipital,
temporal.
same names. I mentioned that we were learning that it's the same names, but the cerebrum's lobes, four main lobes. There are smaller ones, but these are the four main ones. Okay. So this is a side view, and there are ways of pulling it apart. Side view, it's thicker at the back, posterior. This will be the anterior, thicker front. So this is the frontal lobe, parietal there, occipital at the back. Great because it pulled it, but temporal because it did. So, let's now talk about some of those divisions, because when we talk about the lobes, some divisions of the lobes are. Longitudinal fissures, if you had a hairline, a middle parting, it's basically similar to that. It's going to be right to the side between the two cerebral hemispheres. So, longitudinal fissure separates the two cerebral hemispheres. Then we have the transverse fissure. Which direction is transverse? Which plane is transverse? Horizontal, right? So basically, it's going to divide the cerebrum and then the cerebellum. Okay, those are the big two that we have named. Technically, there's a third one, but we use sulcus for that because we want to use the same terminology to divide the lobes up. Right? So the central sulcus separates frontal lobe and then the parietal lobe. Ignore what it says prior to that for a second. Okay? So it separates the frontal lobe from the parietal. Okay? So this is, let's say this was anterior, so this is frontal lobe. Okay? So what is a sulcus? What is a sulcus?
A shallow groove.
Shallow groove. Okay? So this one provides same kind of thickness. Central sulcus separates the frontal lobe from the parietal lobe. Everything from this forward is frontal. So this the central sulcus. So that's a groove. On either side, we have two ridges. Okay, ridges are called gyri. This is the only two we are responsible for. But very specific and very different in function. Okay? So, if I was to say, let's prepone our lab exam, what do we do before you start shouting and crying murder? What would it mean? Prepone versus postpone. Move it forward. So it's in front and postpone is back, right? So this one is called precentral gyrus. Okay, this one is called precentral gyrus. While this one is post-central gyrus. So this is anterior frontal lobe, posterior they are very distinct functions, very important. This is going to be for motor, this is for somatosensory. So that's central sulcus. The other one is lateral sulcus, which technically it's a fissure, it's quite deep, but we just use the same terminology. This way we know it separates another two groups. So the third one, which is the parieto-occipital, kind of obvious, right? This one from the name separates the parietal lobe from the occipital lobe. While the lateral sulcus is just above the temporal lobe, so it separates the temporal lobe, frontal, and a little bit of the parietal superior. So these are 4 and these are 5 grooves. For lecture, we need to know in lab. We just need to know 3 of them. Be able to identify longitudinal fissure, be able to identify central sulcus, and then lateral sulcus. So let's talk about the central sulcus. The significance, it separates the primary motor cortex. That's going to be found in the precentral gyrus from the somatosensory cortex found in the postcentral gyrus. Okay. This is primary motor cortex, and this will be the primary. It's a little bit of a mouthful. Somato. What does somato mean? Body sensory. Right, coming towards the brain sensory cortex. So primary motor cortex is found within the precentral gyrus. So when we initiate our voluntary muscle movement, it's coming from the precentral gyrus. Okay. While the postcentral gyrus is primary somatosensory cortex. This will receive the information from our body, skin, joints, muscles, tendons. Will send information, sensory information to the brain. It goes to this spot. Somatosensory. Okay. This is why this sulcus is significant. These gyri are significant. Okay, next we're going to talk about other sensory cortex. So visual cortex, if you got hit at the back or you fell down, you feel dizzy, not because of the retina, really the brain cells are not communicating to each other. Because visual cortex is in the occipital. Gustatory, does anyone know what gustatory is? So that it's taste sensation. Okay, so the sensory information. Where are taste receptors? Tongue. Do we have any other taste receptors? We do. Hopefully not. Not in the nose. That's olfactory. It's actually a similar type of receptors. They're called chemoreceptors, chemicals in the air they sense, while the tongue and other structures, oral cavity, epiglottis has some too, but they don't play a role in taste appreciation. Okay? But taste appreciation is the tongue, but others are for reflexes. Whether it's hot, whether some food hits the epiglottis, what do we want the epiglottis to do? So there are some receptors that you probably heard the gag reflex, whether intentionally or unintentionally, when they're trying to look at the back of your throat. Sometimes they use the tongue depressor to do have done or to do what? Hit the back, right? So you have the gag reflex. That's because we do have receptors at the back. But in terms of appreciation for taste, it's on. And then that's in our temporal lobe. Okay. Auditory and olfactory is also in our temporal. Where is the one for sensation from body? Somatosensory information? It's low? Right. Oh, somatosensory. But there are things we call what are called association areas. So, for example, sense of smell, sense of hearing, all these different by itself. But when we put some things together, we need to make connections, right? For example, food, sight of food, smell of food, taste of food, right? All those information kind of need to be matched up. So we do have areas which are called association centers, which gives more meaning to those sense of information, right? For example, if you have multiple things in your pocket, you have keys, coins, can we put our hand in your pocket, and then you can grab which one you want without looking, right? Because now you're giving meaning to those sensations that's coming from our fingertips. So we'll talk about them too. So in terms of somatosensory association, which lobe do you think it might be found in? We want it to be close by the main association, the main area. It's also found in our parietal. Okay. If I said we do have a visual association area, where do you think it's going to be located? In the occipital. There's a little part that does go to the temporal, but it's mainly in the occipital, right? So information goes there, you want to be able to associate that, give that meaning to it. Okay? So we do have different association areas. So what are we going to talk about? Here, it also talks about motor. Here's the plan. Because when we want to move things, you want to say, for example, when walking, right? You want to send information to your right foot, right leg, then you want to send information to the left leg. Okay? So something has to help you plan those out. That's not here. This is what sends it. Planning will be just in front of it. Okay? Sometimes they call it association. Your book calls it association. A lot of books calls it premotor cortex, right? So it's just a thing that helps the motor cortex function. Right? Premotor. And then you might also hear of like coordinating, even riding a bicycle has to do with this information. Of course, we need information, sensory information to come through. They go to different spots, including our cerebellum, so that does play a role in balance. But yeah, in terms of mainly coordinating movements, it's our premotor cortex that's found in the frontal lobe. Next one, we're going to talk about how when we are performing certain functions, one side of the brain has more impulses going. Okay. Previously, here that I want to emphasize, previously we used to think that if you're good at one thing, and that's because that is a task related to one side of the brain, they would say you're not going to be good at the other side, which is completely untrue. Okay. So we'll look at some of the tasks. Okay. So left hemisphere controls language, math, and logic, while right hemisphere is for like emotional, music skills, artistic skills, visual spatial skills. That means somebody who can picture objects in a space, right? So if you're good at packing, you have good visual spatial skills, right? So previously, it used to think if you're good at math, for example, you were left-brained, and then you were not good at music. That's completely okay. if we are good at something, it usually means we were exposed to that when we were young. So our brain develops much faster when we were youngest. That's when we develop. That's why usually we have the same, not always, but usually we have more tendencies to our parents or grandparents who we spent a lot of time with because they exposed us to the same kind of skills that they were good at, and hence you do. It is possible for us to learn new skills as we age. It becomes a little more challenging to spend more time, like learning ANP maybe. But are struggling, we just have to keep learning its progress because learning new terminologies, but then making those connections. It just takes us longer. The older we get, the more difficult it becomes learning new skills. So here, just talking about this again. So if you're doing math problem, it's mainly the left hemisphere that's firing a lot more. There is still right hemisphere that's functioning, but here it's a lot more active. Okay. That's lateralization. There's another couple of important areas in our brains more specific to certain tasks. Wernicke's is an area for language that goes from the parietal to the temporal. Okay, it's spoken as well as written language. Another one, Broca's area, that's motor for speech. For example, alcohol affects this area. Okay, so hence we're not able to speak if we are intoxicated, for example, a little buzz. Okay, so this is for directing the muscles. And which lobe do you think this might be? Found in Broca's area. It's in the frontal lobe. Frontals for motor, others are sensory information. So this is also found. Broca's area is also found in the frontal lobe, in front of the premotor cortex. So here is primary motor cortex. It's really two and a half gyri is the premotor cortex, and then in front of that, at the base is where perhaps we don't need exactly the location, but in the sense part from the lobe. So any injury to it, they may have difficulty speaking as well. So prefrontal cortex, that's in front of the motor cortex, premotor cortex. This is what develops the most in childhood as well. Okay, and when you're born, it's the smallest or underdeveloped part. Okay, so our intellect, cognition, recall, personality are all in the frontal lobe. So the injury to it, yes, it's true. People who are suffering with either traumatic brain injuries, strokes, their personality would have changed. Okay, it's because the damage is there. True. And then we'll talk about the limbic system. Has anyone heard what's the function of the limbic system, or another name given to the way it functions? Sometimes it's called the reptilian brain. Okay, that does not distinguish what's right and wrong. Hungry, okay, you got food, I'm just gonna. Okay, so it's our frontal lobe, our prefrontal cortex, which keeps in check. No, that's not socially acceptable. It is their persons who will ask permission if they want to consume, right? So they do have some social interactions that develop this aspect. So it does social. I want to give you this example of eye contact, right? In the US, in lots of countries, if you do not make eye contact, people think that it's kind of disrespectful, right? In some countries, some Asian countries, especially if there is a hierarchy, if you will, in terms of your job as a supervisor, you're not supposed to. It's considered disrespectful, challenging. So they actually look at that. Right? So just note that it has to do with social feedback. So certain aspects of our life develop because of this. Basal nuclei. It used to be called ganglia. We don't use that anymore. So old textbook still has its basal ganglia. Basically, we use ganglia now for collections of cell body in the peripheral nervous system. So lab people, did we hear about ganglion anywhere today? Where was that? Dorsal root ganglion. That's a collection of cell bodies. The sensory neurons, collections of cell bodies outside. So it's wider. So that was used before as a collection of cell bodies, but we differentiate nuclei in the central nervous system, ganglion, ganglia is plural, in the peripheral nervous system. So basal nuclei are collections of ganglia, collections of these nuclei, collections of cell bodies within the central nervous system. That's not actually more specifically within the brain, the cerebrum. That is not part of the gray matter on the outside. So we talked about if I was to kind of draw, if I put gray matter this way, you have white matter more centrally in a cross section like that. You have spots of gray matter in our cerebrum. That would be called basal nuclei. Perhaps we don't need to know all the names, but what it does, and more importantly, when it's not functioning properly, what illness is that caused from, or what illness results from. Okay. One of the other ones I want to connect is a region in the midbrain called substantia nigra. Okay. Basically translate to black substance, because even without any stains it looks black. What's significant about this space or this area is it produces dopamine. Okay. Do we know of an illness that has decreased dopamine and then we give them dopamine as medications? Levodopa? No, not heard of it. Levodopa. Not for dementia, but for Parkinson's. Okay, that's because it's an effect in the basal nuclei. Finally, their number or their dopamine levels are low, so we give them that. Again, it's spread throughout. So the basal nuclei are in the cerebrum, and then we have some other nuclei which together it works as one unit. So here are some functions of it, and I mentioned the imbalance results in Parkinson's disease. So Michael J. Fox was one of the rare cases where he got diagnosed and he suffers with it much earlier on. Right? He was in his late 30s, if I'm not mistaken, when he got diagnosed. Usually this is an illness that shows up 50s, 60s. Muhammad Ali also got it earlier, but he had a different reason. Over this is lots of knots just got disconnected. So if you see a patient Parkinson's, they have no or they don't express their emotions, called aphasia. Okay. They also have tremors. What tremors are they? Anybody seen Parkinson's patients? It is systemic. Is it when they're moving or at rest? It's at rest. It's actually at rest. They're called a pill rolling tremor. You'll see their fingers do that. But when they move, the tremor actually decreases. When we're anxious, we actually have tremors when we're moving as well. So you can differentiate in that sense. It also has those, so it slows down, and then you'll see their gait. How do they walk? Have you seen anyone? They actually walk shuffling gait, it's called, because they do not have enough information, or they're not processing the information, so it does influence their muscle movement. It also, this one normally inhibits antagonistic movements, but in them because they have a deficit or decreased functioning, they have tremors, for example. Emotion, they don't show that emotion. Next one, we're going to talk about the diencephalon. Diencephalon is really three parts. It's the thalamus, hypothalamus, and a region called epithalamus, but we'll just concentrate on one part of it. This is almost correct. It's a little bit too much red here. It should be more orange. Okay, that will be mainly the thalamus. Oh, I didn't mention this. This in lab we looked at it. What was this big structure? Corpus callosum. That's the only connection between the two cerebral hemispheres. The middle of it. Otherwise, the two cerebral hemispheres are separate. that's the only connection between the two cerebral hemispheres. So the diencephalon's lower down, bulk of it is the thalamus, okay, 90% of the diencephalon. Here it's basically, I think of it like the Grand Central, okay, things come in, things leave, nothing really stays there, but there's a lot of movement of information that passes through this area. So most sensory information, olfactory does not, but most sensory information passes through this, most motor information passes through the hypothalamus. The next one is hypothalamus. This is a very important structure. Okay? It's a small structure in terms of size, but it has a lot of nuclei. Nuclei are in collected cell bodies. They have a specific functional significance. Okay? And below the hypothalamus, we have a little, looks almost like a fruit hanging down, is our pituitary gland. Okay? That connection is called the infundibulum, and we'll talk about the pituitary gland more in chapter 16, I believe. We have two parts to it. We call it posterior pituitary and anterior pituitary. It's really only the anterior is the only true gland. Posterior is a storage. It's a storage of from Now let's talk about some hypothalamus functions. Have we heard of any hypothalamus functions? Temperature regulation, yeah. Our body's thermostat is found in our hypothalamus. Anything else? It also produces hormones which control the pituitary. Our hunger, thirst centers are found in the hypothalamus. We also have the autonomic nervous system controls, and the master control is the hypothalamus. Different parts do control small things, but we'll talk about this. Probably not today. I'll finish with this, please bear with me till we finish the answer on brainstem. We'll talk about how each part, for example, the medulla oblongata has the cardiovascular centers, as the respiratory centers. But here is the master control. So it's the control of autonomic nervous system. It does produce hormones which basically either inhibit or will stimulate pituitary gland hormones, or we do have two that's produced by the hypothalamus, stored in the posterior pituitary gland and released from there. We have our eating, hunger, thirst, and even satiety. What's this, satiety? Yeah, feeling of fullness, right? It does, the fibers that come from our stomach, it's called the slow fibers. How quickly does the brain know that we are full? Actually, it takes a little while, which is why, what's the recommended food? And most of us, I'm guilty of this too, but usually don't eat in that recommended time, eat much faster. What's the recommended time for consuming a meal? Yeah, probably not 2 EMTs.
15 to 20 minutes.
It is 15 to 20 minutes. That's the time that it takes us to slowly consume, as well as the fibers to let your brain know that it's full. So if you ever go to a buffet, please do not quickly binge in the first 10 minutes. 90% of the time you will overconsume. Just do not give the time the brain to receive that information. Body's thermostat. We also have our circadian rhythm, which if you do night shifts and you change, that gets messed up. Okay, it takes about a week for it to actually take, sometimes quicker. I remember initially we had to do 7-day nights. By 3-4th day, you kind of get accustomed to it. And then they changed our routine. They said, no, three nights on, and 4 days off. Like that, it was like weird shifts. So like we just got used to it, and then we have to go back to it, which is horrible. It does take time for us to big cycle to get reset. Some people do it much easier. They can, they can just, but generally we take long. It takes a week to adjust. Limbic system also is a little part of it. I would save the emotional part of the limbic system. So we'll talk about that on Monday. But that also plays, hypothalamus also plays a little role in that. So epithalamus, it's supposed to be on top, it's really behind, more of it is behind. The most important part is a structure called pineal gland. Pineal gland produces melatonin. What does melatonin do? Yeah, help us to sleep. So if you had a normal cycle, again, I know I'm talking to people who have changes in their sleep cycles, but if we sleep at the same time, roughly how long before is the peak in melatonin naturally in us? Somebody knows? It's actually about an hour before, 45 minutes before is actually peaks. Okay? So this helps us to feel sleepy, doesn't put us to sleep. So if you do, again, I'm not recommending. If you do take melatonin sleep, don't take it. If you want to be asleep at night, if you want to be asleep at 9 o'clock, don't take it at 8:55. Okay? You want to take it more at 8 o'clock because it has to be absorbed first and it had to peak before you fall asleep. Okay? So that's the most important part about the pineal. Most important one. Epithalamus is the pineal gland, which in turn produces melatonin. Any questions? I think that's a good place to start. Please connect your lab